In this video, we will cover the onset of pancreatic cancer and various therapeutic approach. Though we have covered the top part of our figure in our previous videos, we have not covered this bottom part, that is early detection and diagnosis and therapeutic implication. So when the pancreas is in a healthy state, they are usually considered as the echinacea, and when this echinacea to ductal metaplasia occurs, the onset of pancreatic cancer begins. And one of the major mutations that has been identified in this early stage pancreatic cancer cells are these CRAS mutations, and these CRAS mutations can be inhibited by this microRNA 126 and MGMT. This has been identified. So once the pancreatic cancers this CRAS mutations begins in this echinacea, this ADM transition occurs. That is echinacea to ductal metaplasia, and after that the further changes occur, such as this P16 inactivation occurs, and the cells will undergo this pen 1 stage, and and with with time there is a further mutation occurs that leads to this pancreatic cancer to go into this pen N2 stage, pen 1 and 2 stage, and thereafter mutations such as P53 mutations and SMART4 inactivations at the same time the uncontrolled growth rate will leads them to this pen 1 and 3 stage, and finally it will leads to the aggressive pancreatic cancer stage, which is usually known as PDAC, that's pancreatic ductal adenocarcinoma. So during this stage, there are several different factors that has been identified that could be the possible therapeutic targets or can be used for this early detection and diagnosis. So the major reasons why this mutation occurs is because of the methylations either in the DNA or in the RNA. So in the DNA, the methylations occurs that will lead to the histone modifications, and these histone modifications can be used as a therapeutic implications, or by targeting those histone modifications we can distinguish the tumor cells from the healthy cells, and can target those cells where these type of histone modifications are identified, such as the histone methylation, histone acetylation, or histone ubiquitations. So this chromatin remodeling occurs that can be one of the signature things that can distinguish healthy cells from the normal cells, and this can be one of the possible therapeutic targets, and some of the inhibitors are being used to target these histone modifications, such as BET inhibitor, HDAC inhibitor, E2H2 inhibitors, or DNMT inhibitors. The other kind of modification that occurs is once this RNA is transcribed from the DNA, the RNA methylations might also occur that will lead to the different translations of the protein, and the proteins can acquire because of the different translation of the proteins can acquire different structures, and that will cause different post translation modifications such as different phosphorylations. And apart from this, the methylations also cause this splicing alternative splicing of the RNA and affect the stability of the RNA, and that will lead to the generation of several microRNAs, and these microRNAs can be used to detect whether these microRNAs can be used as a biomarkers that can indicate whether there is a specific kind of whether there is a pancreatic cancer's progression is going on within the patient or not. And some of the microRNAs are being listed here, such as microRNA 21, 25, 210, and others respectively. And there are three types of RNAs are mentioned here. First one is this microRNAs, long LNC RNAs, circular RNAs, and DNA hypermethylations in plasma. So this is related to the DNA, and that can be used as early detections, and uh, can help in diagnosing these pancreatic cancers at the early stage. So this is a brief overview, you can say a revision of how the pancreatic onset of pancreatic cancers occurs and what are the possible changes that occurs in the DNA and RNA that can be used either as a early detections or as a therapeutic implications or for developing some therapeutic medicines to target those modifications. So if you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.